Well, I have to say, I had a student that tagged along, and she was in a class, so I was being paid for instructing her. So I felt I would be double dipping if I also paid for my services here. Besides it, who wouldn't love to come up every 2 weeks and walk through this property? I mean that's a no-brainer right there. Usually, one is curious as to how one gets into the idea of taking the vegetative world and putting it in 2 dimensions and pushing it into a metal cabinet and closing the door. You know, what would possess you to spend your life doing that? And I guess I have to say it all came about because the town I lived in Wisconsin had a leash law. And my responsibility was to walk the dachshund, okay? Well, you can't get very far away from a tree when you're walking a dog, right? And the dog is sniffing at the base of the tree. Now, there are 2 things you can do. You can look at the dog, or you can look at the tree. Well, I looked at the tree. And so it was a sort of it became a terminal addiction. You know, once you know one, you want to know another, and then why are they different? And you know, it's terminal. You just can't quit. So that started the whole process. I started out my academic career in medical technology. That lasted about 6 months. The anatomy class was full, so I took the botany class and never looked back. So all of those serendipitous events led to this point in time, to a captive audience and to a collection of dried, brittle specimens. We are actually at the very end of the era of collection. Began with the era of great discovery, when new vistas were opened up by sailing ships plying the oceans and discovering whole new floras and faunas. And so one of the books I brought along is about the age of discovery, and it talks about how, yeah, that's fine, no problem. It talks about how collections began. People with deep pockets had thought it very fashionable to have these bizarre collections of plants and animals. And so the first zoos were actually came about by people paying naturalists to go out and collecting in the wild kangaroos and sloth bears and bringing them back and housing them in cages. And the more bizarre your collection, the more prowess you had in the community. And so too with the plants. Gardens wanted new and different and the most exotic possible, and so these plants were brought back, both by seed and also by collections. Artists went with the naturalists frequently. The naturalist was also an artist, and so the color of the flower and all of that was preserved not in the specimen, but in some of the most beautiful drawings of the flora were done at that time. Today, herbariums are out of vogue. The university at Charlotte and in Nashville have terminated their herbarium. They are now housed, one was just unceremoniously thrown out and another was put in a nature center. Others have been delegated to hallways, as they have in the University of Wisconsin. And they are used less and less. The idea of the whole plant is largely gone. The research now, you can't get money to fund a herbarium, to even buy a herbarium cabinet. The science foundations no longer support those endeavors. So we have collections, very valuable collections throughout the world that are being maintained, and a few, like the Missouri Botanical Gardens, are still adding to the collection, but no new herbarium are truly being developed. Now is the age of DNA, and one person put it to me very succinctly. He said, well, if you got a plant, you're only showing the genes that are being expressed, that are actually, and they aren't, it's only about, what, 10% of the genetic makeup of that organism. So you're only looking at 10% of the data for trying to figure out how it's related to another organism. But herbariums are incredible reservoirs. The one here, you're probably in the smallest herbarium in the world. Without a doubt, probably one of the smallest. But the plants that are there reflect the plants that were here on this land when it was first set aside. So that in 50 years, as the area develops, as you can see how the flora is changing, what is persisting, what has come in since then. So you have a baseline for all future observations here. Not only that, with the collection, you have information. This is climbing fern, Lycoglia is the genus, and it is found only at one spot in the preserve just below the little pond. Lycoglia is a noxious weed, not this particular species, but a very close in Florida. The climbing fern is the common name of it. So that it's going to be very interesting as this area remains preserved. How is the Lycoglia, how's the climbing fern going to, its distribution change? You have documentation where it was. Now in 50 years, it'll be very interesting to see how it's spread. Whether it too is going to become a noxious weed or not. So that's just one example of how and this specimen, by the way, if you looked at it 200 years from now, it'll look exactly the same. It'll still be in 2 dimensions, but it will still have the information down here of where it was found, when it was collected, who collected it, who identified it, and all of the particulars that go along with identifying this particular species. I'll show you how this is done in just a little. With the collection, you have both the underside and the upper side of the leaf. You have the fertile leaves and the sterile leaves, so you have all this information encapsulated in 2 dimensions. Halesia, which is silver bell, has flowers and fruits, so you have both of. But the interesting thing about the Halesia here is you have some very, very old specimens, and all the books say the flowers are white. Yours are pink. Yours are pink. I've never seen that in any of the books. White flowers. But maybe when they get very old, are they old specimens that become pink? Or is it a variety of it? You know, to be discovered. We have fruits here. Inside the fruit are seeds. So that if that becomes extinct, you can come back here, get the seeds, and they will be viable. So you have a reservoir of the possibility of reintroducing something should it have no longer become extant on the property. And yet, you also have an endangered species. Well, you have actually 2. I haven't got the ginseng yet, but I will. But yeah.
Were those leaves eaten when you preserved it?
Yeah.
Not after, okay.
Yeah, that's a good, I'll go back to that question because that's another important piece of information that a herbarium specimen. This is butternut. It's closely related to black walnut, and it has some differences. Both black walnut and butternut have a chambered pith, and you can see that here if you wanted to look at it. They both produce the nuts, but this is a short-lived tree, and walnut is a long-lived tree. This one is being negatively impacted by a fungus, so that in the populations in the wild are disappearing. In the secluded valley of Walnut Creek, some of these are still producing fruits. They're still actively growing and providing a seed source, perhaps for reintroducing it once this fungi has progressed. So here we have an endangered species. Here we have a species that's found only in one location, but what's going to happen to it in the near future or the distant future? All this information is available. Leaves are being eaten. Okay, so you know that there's an insect or something is that insect going to become more and more prevalent? I worked in developing 13,000 specimens at the University of South Carolina in Spartanburg at their herbarium. And one day I had a phone call. They said, "Do you have this particular plant?" I said, "Yeah, I got it." "Did you get it in a cotton field?" "Yeah, it's a weed in a cotton field in Cherokee County." And they said, "Could you send us the specimens?" I said, "Well, this is from the University of, I think it was Alabama." I've slept since then, so don't hold me to it. And so I sent the specimens, and they discovered that there was a fungi growing on this weed that was growing in a cotton field. They harvested spores from those, from the fungi, and they used it as a biological control of the weed in the cotton fields in Texas. The thing that's happened is that they were using herbicides, and herbicides tend to be fairly broad spectrum. And what happens is that the weeds that grow in the cotton fields were very close relatives to the cotton. So any herbicide that would have affected the weed would also have affected the cotton. So they couldn't use, but this fungi, which was very specific for the weed, could be harvested, could be used, and the spores of that fungi would be a natural deterrent to the having to put more and more herbicides on the cotton fields. So you just never know what's going to be involved in this bit of information, you know. You have it preserved, you have seed source, you have diversity information. There's an incredible amount of information locked up in those little cabinets there in the two-dimensional world of the herbarium. Herbariums have been developed for a number of reasons. Here, it's to document what was here at any point in time. The largest herbarium in the world is in France. It's almost 10 million specimens. And it was developed in the age of discovery, and it has collections from around the globe. The first eucalyptus ever collected was housed there. Kew Gardens, similar environment. They have only 7 million. You know, France is a little bit ahead. The Missouri Botanical Gardens, I think they're at 7 million too. Their specialty is tropical flora. And they have stations down in the tropics where they are monitoring changes and documenting what was there, looking at future. Sort of, we've got a mini Missouri Botanical Garden right here. Okay. Right. Probably the most interesting one is the Russian one. The Russian have 7,150,000 specimens, and they were largely collected in the last 200 years. And one of their leading geneticists, Vavilov, became very, very concerned about the loss of genetic variation in crop species. He analyzed that 90% of the genes in apples have been lost by domestication, because you grow the same thing day after, and in one field is just exactly genetically identical to the next field, so you get the crop growing at the same time, the same color of apple, the same. So all of the genetic variation has been bred out. And he realized that we don't even know where the wild apples came from. So not only have we lost this genetic variation, we don't even know if we can get it back. So he started going around the world and trying to locate areas where the crops were domesticated and collecting the wild plants in the vicinity so that that genetic variation could be preserved. He collected specimens, he collected seeds, and he went around the world, and this is where he made the mistake. He went to Arizona. And he told the people in Arizona, "You're fools. You are absolutely missing the boat here. You've got so many different food sources that the Indians used that you've just not dealt with, you know. We live in a world of 20 food species, half of them grasses. There's so much more food out there, but we're not taking advantage of that." He said, "You don't even know where these plants came from that you're using today. Where are the wild tomatoes? Where are the wild potatoes? Those are your plants, and you don't know diddly squat about them." Okay, I mean, he was pretty tenacious, and he did that in around 1932. Stalin heard about his fraternization with the West, and he cut off all funding. He put Vavilov in a gulag, and his assistants were deprived of jobs. What happened to those seeds? Famine was rampant in Russia, particularly at the early part of World War II. All of these seeds were stored: rice, barley, oats, and his people were starving. Did they eat the seeds and survive? They buried them. They buried them, and some places we don't even know where they buried them, but they preserved them, and many of them today are in the international seed reserves in Sweden. It's a fascinating tale. What did he do wrong? He was telling us we had our heads in the sand, but Stalin only saw that he was fraternizing with the West. If you want to read the story, and it's a really, really it's where our food comes from. It's the story of Vavilov and his exploits. This is, I don't even know, but I've got half of it underlined. So that's how important I think that particular book is. So that's the Russians. So herbariums can have a very specific role or they can have a generalist role. In the Midwest, most of the herbariums in Iowa, for example, are grasses. Well, there's where do you think many of the seeds came back for planting the shortgrass prairie and the tallgrass prairies?
From those collections.
From those collections, and from areas where they identified they were still growing and could go back to that area and see if it was still there. Now, grasses are really fascinating, and they are so important to us, not for golf courses. That's not what I'm talking about. What I'm talking about is oats, barley, rice, corn, millet, all are grasses. They are our starch source. Sugar. It's the most important food group we have are the grains, and all of them are grasses. I don't advise you to go out and eat your clippings, but they are definitely worthy of having a reserve, so that you can reestablish a whole ecosystem based on these reserves and knowing exactly where they grew, because you have that data on the sheet. What I want to caution you about is that there are some problems with herbarium. They're costly to maintain, they take up space, and they bias you in certain respects. Many collectors will tend to, particularly students, they'll take the smallest because it'll fit in the press better, right? So the specimens in the herbarium may be a diminutive form of the wild type. The color is largely gone. The smell is gone. And there is a tendency to over-collect the pretties. All right? So, you're not going to have a lot of tourists going in and looking at the grass collection. It's just not going to happen. And you're only preserving the physical evidence. All the DNA, but you are preserving the DNA. So you do have an encapsulation of the DNA within the seed. And so, you do have that information preserved, but it's not a visual imprint. But the specimens are as permanent as you can get them. The paper is rag. It's old cotton rags, 100% cotton fiber. It's pH neutral, so it will not yellow. It will not become brittle. It will stay in this kind of off-white color for perpetuity. The glues are water-based. Why would you use a water base? Well, the plant's water-based, so it will go into the tissues. And if this should become damaged, you can take it, put it underwater, and float the specimen up and remount it. We use bookbinding tape, the cotton bookbinding tape, to strap it. In Russia, they use silk threads. They have little old ladies that sit there and tie knots underneath, and they're beautiful specimens. I mean, my God, the time. Well, I didn't have silk thread, so I used that. The label is cotton fiber. Now, here's a problem I encountered. How do you print the label? You can't use inkjet printer. There's no evidence how long that ink will last. So you have to use the whole carbon, you know, the ribbon of the old typewriters, or the Apple image writers. They use the carbon ribbon. You know the carbon's going to be around. But, you know, these are the sort of things that I called Hewitt Packer. How long are your inks going to last? Well, how long do you want them to last? I said, oh, 500 years. And they go, we probably will. That's not good enough. Now when you're spending 2 hours to 3 hours on a specimen, you know, and you want to have some evidence that it's going to be around a while. Those are some of the problems. So let me show you as we progress through here. It's an 18th century science, maybe 17th. It's largely unchanged. This is a plant press. Well, it looks like any other plant press, except I made it myself because I wasn't going to spend $50 a plant press from Carolina Biological Supply. And it's basically just a way of, so here we have, so that they lie flat when the straps are, this adds even pressure, and then you keep them tight, and it'll dry down into paper thin collection. This is just, I don't know what they did before a coronated cardboard, I really don't, but this acts like little chimneys. So that when you put it on a heat, oh, and I'm so glad he did this. So you put it on the heat, the heat comes up here, dries the plant, the vapors. You just don't want to make sure you just don't keep turning it back and forth, so that, because when the poor water bottle cools, going to go back and forth forever. So this is a ventilator, is the piece of corrugated cardboard. You can buy newsprint that doesn't have anything on it, but I just used the regular. And you don't want to have a lot of ink on it because the colored ink don't absorb water very well. So, and then when you put your specimen, you put the date, you put where you got it, so the information is there with the specimen. We have blotter. This used to be very common. This is when they had used the real pens and you had to blot the ink, you know. But it's still used in a variety of archaic functions like herbarium sheets. So you have alternation of these. And so when you're in the field, they used to have things called basculums, which were cylinders with a little trap door in them, you know, and you walk down there like that. They weren't heavy, but they were awkward. See, so the bag, you can just walk down the road like this, you know. The briars aren't going to bust your bag, you know. It's really important, though, when you go in to get a drink, take the bag off. People just really don't understand that sort of attachment. But a white bag, if it's a really hot day, you might put a little water in it, a couple of drops, but most of the time, just that will do. Now, specimens, we can start with the easy ones here. Maybe. Okay, well, so this doesn't look very pretty, but it has some valuable, this is black cherry. And you probably notice in spring when it has the white flower, but it has fruit right now. So that's a very important piece of information. When do the fruits ripen? What sort of environment do you have plants producing fruit? Just because it does flower doesn't necessarily mean it's going to produce fruit. So you take this and you arrange it on your, and you want to arrange it in such a way, now why this is exactly the same size as your herbarium sheet. So you know, and so you say, well, let's see, maybe I don't want all that, so I'll just, that's the vegetation. Make sure you have the two sides of the leaf, spread it out so that it doesn't look like you're hay baling. That's what we call it when you just stick it in and, it doesn't look very good. And then here we have the fruits. So we make a second one with just the fruits. And you can arrange it so that you have bark. You can take and put it in like that so that you can arrange it. With each one, you put the information on the side, so that's absolutely contained it with it. Some things you can put the whole plant in, like this one. This is a little plantain. It's blooming right now, but you probably don't notice it. And we can put several in so that they show, this has the fruit, this has the flower on it. We have the roots, so we know how much soil is taken up by where it's collecting its nutrients from. And this one's nice because the leaves are linear, so you don't have to worry about it. That'll make a nice collection right like that, 2, sometimes 3. If it's an important one, you can make an additional one right next to it without the paper, because you have that information, you can tuck them in together. What about something, is trumpet creeper, so that we'll have fruits, we'll have buds, and we'll have flowers. So we'll have all of them in that collection. We'll collect it so that we have both the top and the bottom of the leaf, and also noting that it's a vine, so that you know the pattern. So this one would be something like, and then you can bend it up, or you can use, I often, if I've got enough sheets, I'll do like that. And this takes time, but unless you do it carefully, all you've got is a bunch of hay when you're done. It's about as valuable as a bunch of hay, and you can feed it to your horse, but other than that, it's of little use. So, if it's a tree, you have to note that it's a tree, because you aren't going to take the tree. I mean, you could, but you would spend all your life on one specimen, and it doesn't work very well. So you take the leaves, you may take a little bit of the bark, the twigs, so that you see the terminal bud and the shape, and the year-old and 2-year-old bark, so you get as much information as you can. Sometimes you have challenges. This happens to be sumacs. What in the hell are you going to do with that thing? It's not going to fit. Well, what you can do is you can take a single leaf to represent the leaf, and then once you've done that, then you can say, cut this off or bend it back, and cut that off, maybe cut this off. What you want to make sure, though, is that you have both the underside, and you can fit it like that, and then it will work fairly well. So it takes some ingenuity. Bending them in W's and U's and V's, and any port in a storm to get that information. And things like this don't frequently, so you take a photograph of it, or you take and cut it in half. Opuntias, prickly pears, and cactuses in general are tough nuts to press. That's kind of a mixed metaphor there, isn't it? But what I discovered with Opuntia, thank God we only have 2. You can take a surgical steel knife and cut it in half and salt it. Salting the fleshy parts will then allow. I've had Opuntia flower in the press. You know, it dries, didn't care, it flowered. So you have to kind of desiccate it, and salt works well. I haven't seen that in the literature, it's just something that I figured out how to do. Yeah.
So you desiccate it, you reduce the salt before you put it in the press?
Yeah, just salt it. Yeah. But there's another problem though with a herbarium specimen. Fleshy items they shrink so that they're undersized, you know? And so you need to get some information about and indicate that this was fleshy, treated with salt or something like that, so you have a record of how you went about that activity. So each and every, there's no one right way to do it, but if you don't do it in an aesthetic fashion, and if you don't preserve as much information, you're just hay baling it. If you've got and you've got pieces sticking out, or I could take a look at a herb and say, this guy's not going to have anything, you know. If he's got a clump of grass, rather than taking it and separating it out, you know. If he's got a fruit like that, I might put it in a totally separate press so that it doesn't affect the quality of the other specimens. Once this is put together, you cinch it up. Actually, this is really good if you have a dog as an assistant. You can get the dog to sit on it. I actually have trained several dogs to do this. And you gotta watch their tails. They really don't. But then and you can still buy these. You can you can use the military belts. You just 18th century. You could have done 15th century. Except maybe corrugated cardboard. I don't know what you would use, but then just extra blotter paper or something like that. Okay. The tighter it is, the better. And another thing that's going to ensure a bad collection is if you don't come back and tighten it up. You take this over on your wonderful plant dryer, you put it on, and there she goes, up. And how long does it take? Depends on the number of specimens. It depends on the heat. Got to watch the heat. I had a friend who was doing a Flora of Alaska, and he was out in his canoe, and he set up every night Coleman lanterns, and he put the plant presses over the Coleman lanterns, and it was a really bleak, drizzly day. So he put a tarpaulin over well, it got so warm under there, he caught all his specimens on fire, and he lost his whole summer collection. So, I mean, you just have to learn by trial and error on what makes a good specimen, what doesn't make a good specimen. Then you just take another expense is largely in paper, because a herbarium sheet will run you 22 cents a piece. And it's getting higher because the rag content is harder to get. You know, all of our T-shirts are going to the third world countries now, and not going to the rag bins for making paper. So that's the sort of art. My father and my mother were artists, so I think maybe one of the reasons that drew me into taxonomy, into herbarium specimens, is it was the most aesthetic of all sciences. They made me take, most of people would say, you gotta take something practical. Go take biology, take physics. My parents says, you gotta take art. I had a minor in art as an undergraduate, you know, and so I was kind of programmed to take and look at the more aesthetic sciences, and maybe that's why I came into this detritus world. Couple of things is that everyone wants to know what a plant is. And what the herbariums will do is that you say, I know it's a damned aster, you know? So you can go in there and you can lay out all the asters and you can say, oh, this is what makes this species, this is what makes this species, this is what makes this species. And you also use these identification books, and some are better than others. One of my favorites deals only with trilliums, but it is incredibly detailed about trilliums. 7 years to bloom, seeds distributed by ants, very, very localized in distribution as a result of that distribution pattern. The seeds, oh, these are so cool. The seeds have a little food packet right on the, called an eliosome, if you really want to know the name. Don't ask me to spell it. And the ants, that's food for the ants. So they carry it back to their nest, take it down there, the youngsters feed off of and then they take the seeds and remove them. So they're scattered around this old ant mound. The water trickles down. There's, I'm sure I've never analyzed the growing value of ant shit, but I mean, there's got to be some nutrients there. So I mean, so there is, you know, they're all in little mounds. I mean, who would have thought? But it's one of the reasons why trilliums are so endangered right now, because our native ants, which are doing the distribution, are being outcompeted by things like fire ants, which are non-native. And do you realize that 40% of our spring flowers use this mechanism for seed dispersal? Just think of what's going to happen. 40% of our spring flowers without a means of dispersal. Just another thing to keep you up at night. If you want to look at plants via communities, there are two that I recommend. Forest plants of the Southeast, very, very, lots of information, not a large number of species, but the common ones, the grasses, really nicely done. This one is a little bit more detailed and deals with more communities than just forests. But my favorite one, and Dan's the one who showed me about this book, is this one, The Wildflowers of Tennessee and the Ohio Valley. Now don't read the cover. You'll never open it, because you say but it works beautifully for here. And the thing I like about it is it groups say you know it's a rose. Well, it's got the roses grouped together. And then it has the common ones beautifully illustrated, and then it tells you some of the more rarer ones and how they differ. So it has a diversity of plants that the others don't have because of that. And it gives you a lot of, and it's not expensive. And it's designed to take in the field, as opposed to the Flora of the Carolinas. 1968, I think it was. It's incredibly detailed and incredibly out of date. All of the names have been changed to protect the guilty or the innocent, I don't know. So how do you deal with that? They still have some of the best illustrations, though, of and range maps. Remember, there's a lot of dots that aren't here because these are 68, which is, whoa, that's a long time ago. So you can go to the North Carolina Botanical Gardens website. I'll just show you. I can't show you because you don't have, but you go to my website at the university, USC Herbarium.org, or just USC Upstate, and go to the Herbarium site there. So there's two ways of getting to it: USCUpstate.edu or USCUpstateHerbarium.org. And I'll show you what, and this is the 2008 version that is no longer available in hardbound. There are no illustrations. There are nothing to help you, okay? The reason being is the old birds that wrote this have not released the drawings. So they're now producing it free of charge, but without drawings. Well, but there's still, the University of North Carolina Herbarium has the drawings, and so they have in perpetuity. Last night I went on to see their latest edition. It's called the Flora of the Southeast and Mid-Atlantic. Now they've included not only North Carolina and South Carolina, but Virginia and Florida and Georgia. So now it's 1,000 pages long, and it's still a work in progress. They've rearranged it so you can't find the families, and there's still no illustrations other than distribution maps. And the distribution maps are excellent. But this you can get, I have it here in case you want to take a look at it. I have one other thing to show you, and that is something that I'm making available free of charge. And it's, over the years, I've taken a lot of photographs of plants. So, and so I said, well, they're all in a hodgepodge. Why don't we try to organize them? 6,000, 7,000 photos. And they're very informative. What I've done is I've organized them by families, and I've put them on a thumb drive. And you can say, it's a member of the Rose family. You can pull up that file, and it'll have all of them in alphabetical order, and they're named. So you can take a look and see, instead of taking all the herbarium sheets and spread them out to Dubuque, you can do it at the leisure with a cup of coffee. So we'll just, I guess I better tell them I'm here. And so here we go. Are
you up?
No, I'm not really, I'm locked. There we go. Now I'm up. Okay, here we go. So here is the list. No, it isn't. Well, it's a list by alphabetical order of the families, okay? So, there they are! Okay, let's just do with, so I want the passiflowers. Now, I don't have all of them. Come on. Well, let's go with something a little bit more exciting. bought the asters. Okay, so here are all my aster charts of the aster relatives. And so you can say, well, and you can scroll down through them. The C's, the D's, and you say, well, maybe it's this one. And you can, I'll say, well, no, maybe it's, let's just pick. Okay. Here's the fruit, and here's the flower. I'll say, well, let's see what the next one is. And there's the bumblebee that's using this guideline to go around the circle and getting the pollen on its back. Okay? All right, well then let's just see what else. There it is without the bumblebee. Look at that. Isn't that cool? Look at, oh, and then, you can, I think you can blow this damn thing up too, somehow. Well, anyway, you can blow, I'm not going to try it, but used to be there was a...
That makes me want to stop killing those damn carpenter bees.
Yeah, here we go. Here we go. Okay, and then we can move it around. Pretty awesome, huh? And these are handheld photos. I mean, I didn't take them with, so it's a fun thing to play with, even if you don't. Y'all all know sassafras, right? Well, not my dog, the plant. Sassafras is in the Lauraceae. See, if I remember how. Well, I forgot to remember to put it in. Well, sorry about that. This is black gum, Nyssa sylvatica, with the little tiny, tiny flowers in spring. They're separate male and female, so this is a male that has only the male anthers in it, so you can actually see that. So anyway, this is soon to be available. I've got to put the Lauraceae back in there so that and it's a work in progress. But if anybody would like, I think you have that's what the thumb drive. Yeah. Or if you'd like to play with it right now, I think it does kind of bridge the gap between the herbarium and the real world by giving you the color and also giving you close-ups and habitat and pollinator and all sorts of things that well, just one more here, just to tantalize you a little bit. Let's go. Everybody knows the magnolias, right? So here I have a tremendous number of, everybody loves magnolias, but let's try this. I don't know why it's not listening to me. There we go. That's on my wall, so, I mean, the picture is. But you see, there's the fruit. Oh, this is kind of cool. You see that the seeds dangle down and they're bright red. And the reason for that is that they're enticing the bird to eat it. And so that is an actual, and they only come out when they're ripe. So you have all this kind of neat, here's the unripe. Pretty cool. So you get an idea of that it's an art as well as a science if you like plants. But I've told Mary before at the beginning that if PAC would like to sell this and as a moneymaker, I'd be glad to make it available to you. So I mean, I can enjoy them. This is the Virginiana. This is the low, but this actually will grow in secluded areas. Probably not in Polk County, but in Spartanburg. So, but this is I say it's a work in progress. I'll always be adding to them, but I think with you know, you can learn enough to know what the family is or suspect it's, if you suspect it's a rose, we have a few examples. Linkers. So you give a good example of what's possible. Crataegus, that's kind of a cool one. But anyway, if that would be helpful, I'll be glad to make that available to you.